Welcome everyone, and thank you for joining today's Ask the Expert webinar, Is Your Wearable Device Safe? Our presenter today is Blake Darko, Team Leader of the In Vitro Biocompatibility Testing Program at EAG Laboratories. Our moderator is Dr. Philip Furco, Senior Scientist with EAG, and I'm Melissa Campbell, Marketing Manager assisting with today's event. Before we get started, let's go over a couple housekeeping items. All attendees are muted. However, if you have questions during the presentation, please enter them in the questions section located in your control panel. You may send in questions at any time during the presentation, and if time permits, we will answer these questions at the end of today's event, or we will follow up with you soon. With the abundance of pre-submitted questions, we will not be able to cover all of them during today's presentation. However, the team will send you an answer by email. At the end of the webinar, a survey will pop up on your screen, and your feedback is greatly appreciated. And now I will turn it over to Blake.
Awesome, thank you, Melissa. As Melissa said, I'm Blake Darko. I'm the team leader of the In Vitro Biocompatibility Lab here at EAG Laboratories. My degrees are in biomedical engineering, and I originally worked as a product engineer in the medical device industry before specializing more into in vitro testing to characterize biomaterials. So I'm excited to kind of walk you through the capabilities that we have here at EAG and are developing here at EAG with the focus kind of on the wearable industry. So a little background on EAG laboratories in general. When it comes to understanding the physical structure, performance, chemical properties, and composition of materials, no other scientific service company offers the breadth of experience, diversity of analytical techniques, or technical ingenuity of Eurofins EAG Laboratories. We don't just perform testing, we drive commercial success through thoughtfully designed innovations, investigations, technically superior analyses, and expert interpretation of data. EAG Laboratories is a division of Eurofins and has a team of more than 1,000 employees, over 2,500 testing instruments in 20 laboratories around the world. So to kind of kick things off here, I want to walk you through the outline of today's presentation, where we're going, and what I hope to communicate through this presentation. Ultimately, EAG, we want to help you learn more about your product to make more well-informed decisions about your wearables. So to start, we're going to get into just the in vitro testing at EAG, what kind of industries we support, what kind of needs they generally need. We'll define what a wearable is, what's the difference between a consumer electronic and what's a medical device, and where is that distinction lie. What's the risk associated with a wearable? We'll get a little bit into the regulatory and industry framework. I'll give you a little bit of a primer on biocompatibility and how that's assessed, as well as go into some details on in vitro assays. And then finally, we'll wrap it up with kind of a discussion on how to navigate a quote-unquote failed test, and how EAG as a whole can help you through that. So as Melissa mentioned, this is an Ask the Expert formatted presentation. So we received a number of questions from the attendees ahead of time that we kind of incorporated into different slides. And then a few of them we call out specifically to address kind of more like specific questions. So if your question wasn't addressed, in the presentation, we'll get to you with a follow-up email. So real quick, just kind of a primer on the in vitro target industries and needs, who we work with here at EAG and the in vitro lab. So as you can imagine, the medical device industry is up there, as well as consumer products and raw material suppliers. And they're generally going to need one of three things, really kind of the first two. One, they're going to need data to support a regulatory submission. Or two, they're working through different design prototypes, design screening, and trying to select the best material to incorporate into their device. And then finally, we work with some companies on actual lot release testing of their products to ensure that every batch, every lot of product that comes off of their assembly line is going to be safe, and they can market it as such. Today, we're mainly going to focus on the center column, so the consumer product industry as well as medical device, and kind of in the design screening aspect, and we'll talk a little bit on regulatory submissions as well. So, just to define a few terms, I want to walk through, what is an in vitro biocompatibility assay? In vitro is Latin for in glass. It describes a process performed outside of a living organism. In vitro models are designed with specific cell lines and conditions, and they're used to mimic a disease state or a physiological condition. And then assays characterize the biological processes that are undergone, in the model. And then finally, biocompatibility, and this is a definition that I pulled from ISO 10993, which is the ISO standard on biological assessment of medical devices. So biocompatibility is the ability of a medical device or a material to perform with an appropriate host response in a specific application. Real quick, just kind of right off the front, some advantages for that in vitro biocompatibility assays have. One, they're fast. Two, they're really cost effective. And then three, they're scalable and also fairly reliable. So these three kind of distinguishing factors really deliver, a lot of value to investigators looking to characterize their product. So next, I want to talk about what is a wearable. The first industry that probably comes to mind when wearables come up is the consumer electronics industry. So here you have a guy, working on his computer, and if you look, he has, quite a few things that are actually in contact with the skin. So he has a wristwatch on, he has headphones on, maybe a ring. But again, skin contacting products, maybe it's a biometric tracker, maybe it's a fitness tracker that they're wearing, as this person is here on the bottom. And so examples of this are watches, bands, rings, glasses, headphones, etc. Anything that is in that prolonged skin contact duration. The second kind of industry category would be within medical devices, and so this would be continuous glucose monitors, lead electrodes for EKGs, and even braces or orthotics. So think of a knee brace that's in constant contact with the skin. Can be categorized as a wearable. So these are kind of the products that I had in mind when crafting this presentation today. So next I want to talk about what the risks are that are associated with wearable products. And right off the bat, there's prolonged contact with the skin. And a lot of times this is heightened with rigorous activity. There's kind of a gray area in a biocompatibility sense of, a lot of questions we got today are, how do we characterize it as a medical device? Is it a consumer product? So it falls in a regulatory, biocompatible gray area. It's complex from the supply chain side. So especially in the consumer product industry, some supply chains may be less regulated. And I know that we here at EAG have extensive experience, working with clients on finding the right supplier and characterizing, that the material that they're purchasing is truly the material that it's advertised to be, and so making sure that no hazardous additives get incorporated into the materials through the supply chain. So these risks, if gone unchecked, can lead to skin irritation, skin sensitization, and in worst cases, chronic inflammation from excess cytokine production from that physiological response to the toxins that are leaching through the skin. So, what's the risk? Consumer product recall. So it doesn't take long. It doesn't take a long Google search to find pretty substantial product recalls in the consumer product space. And here, is a timeline. It's not only a recent issue, but it stretches back a decade. So in 2014, there was this substantial fitness tracker that was recalled due to skin irritation, which was linked to a high nickel content. 2016, there were 2 fitness trackers that had major recalls due to skin irritation, skin burns, or blisters, and discomfort associated with those products. One of them was a smaller company, and then one of them was a pretty significant company that's not usually in the space, and I don't think they're in the space anymore. 2021, there was a VR headset recall that was due to skin rashes. 2022, another fitness tracker from skin burns. And you know, the first ones that I kind of highlighted are all consumer electronics. The last one here was actually an FDA recall for a pediatric thermometer due to skin burns and irritation associated with chemicals leaching out of it. So ultimately, you know, these recalls lead not only to lost revenue and lost customers, but also damage, you know, to companies' reputation. And that's something that we think can be mitigated with testing on the front end. So now this is a audience submitted question. How do test limits differ between medical devices and consumer electronics? So I want to spend some time kind of unpacking that here. So I mentioned earlier, ISO 10993 is the industry standard for the biological evaluation of medical devices. So to define what a medical device is, that is where we went. So according to 10993, a medical device is an instrument apparatus used for human beings for one or more of the specific medical purposes. So that could be diagnosis, prevention, monitoring, treatment, or alleviation of a disease, diagnosis, monitoring, treatment, alleviation, or compensation for an injury, investigation, replacement, modification, or support of anatomy or of a physiological process. And then a few other ones that aren't necessarily as related to wearables, but supporting or staining life, contraceptives, disinfections, all of this with the caveat that it is not achieved by pharmacological means. So in other words, it is not a pharmaceutical. So most of the difference, well, we'll go to this next slide here, which is a Venn diagram that I prepared, kind of looking at the risks associated with medical devices and consumer electronics and where they overlap and where they are different. So both of them have, you know, prolonged skin contact. They're both impacted by sweat and movement, which could, you know, heighten the leachables that were to come out of a product, or they're used underwater, all of these things. And then finally, you know, they have a risk of a physiological reaction to those materials. So where they differ, the information gathered for a medical device is used to inform medical decisions or clinical decisions, or they're used to treat a condition. Whereas consumer electronics, the data or the usage is primarily for the consumer. So the big distinction comes with, you know, clinical claims. So going back to the question, the original question, which is how do test limits differ between medical devices and consumer electronics? You know, we'll answer the question with another question, and that is, what is the risk? So certainly from a regulatory standpoint, there is a difference, and that's not necessarily our, you know, field to advise on. We're the specialists kind of in the testing. So when it comes to the testing, what is the risk? Well, again, that's the supply chain quality control and the material hazards ultimately leading to product recalls. So if we can control, you know, the first two, we can mitigate the last one. So ultimately, you know, the risk decision is made by a, you know, an SME in biocompatibility or a toxicologist via a biological evaluation plan. But the testing that we're kind of outlining here today can really help, you know, inform those decisions and inform that design decisions. So let's look at a few resources that are available for this, and there's countless resources. First one is FDA. FDA website in general, it has a lot of content that you can sift through. Again, we're talking about materials, but this is just a consolidation of a bunch of guidance documents with digital health content. So a lot of this might be software related, which is applicable, you know, obviously to these products. But when it comes to the material standpoint, it's best to look more towards you know, the industry standards for the medical devices, because those are required endpoints that need to be evaluated. So for that, I mentioned earlier ISO 10993. So this is, you know, the cover of part 1, the biological evaluation of medical devices, evaluation and testing within a risk management process. And within this, there is a table A1, which is screenshotted here, that outlines all of the device categories, the contact categories, as well as the contact duration. So I'll get my laser pointer out. For example, we have a surface medical device that is in contact with intact skin, a mucosal membrane, or breach or compromised surface. And there is limited contact, prolonged contact, or long-term contact. You would then use this chart to determine what endpoints, which are marked by an E, are recommended to be evaluated or required to be evaluated per the standard. So for a wearable, which would be most equivalent to a surface medical device in contact with intact skin for really any duration, so limited contact or long-term contact, you kind of see this 3x3 matrix here of the required endpoints, and this is what we call the big three in biocompatibility. The reason for that is that, as you can see, is that all of these are required endpoints for every type of device, regardless of contact, regardless of duration. The cytotoxicity, sensitization, and irritation is a required endpoint for all of them. So with that, I'd like to kind of introduce our big three, in vitro program that we either have, internally validated or still in process development. But to kind of go through some definitions here, starting with cytotoxicity. So cytotoxicity is cell death in response to a substance or material. The ISO standard for this is ISO 10993 Part 5, tests for in vitro cytotoxicity. And the common method that we utilize is the MEM elution method, which I'll detail here in subsequent slides. The next one that I'd like to talk about is irritation. Definition of that is localized, nonspecific inflammatory response to an application of a substance or a material. The ISO standard for that is 10993 Part 23, and the OECD standard, which kind of informs that, is 439, being the method, the RHE tissue model, which stands for reconstructed human epidermis tissue. And that is actually a skin layer grown, in vitro in the lab to mimic how a skin irritation performs. Finally, I'd like to talk about sensitization, and this is an immune response mediating a skin reaction to a substance or material. So it's a larger systemic response. The ISO standard is 10993 Part 10, and some of the OECD standards which outline it are OECD 442 C, D, and E, as well as the other ones. So 442 is all sensitization. The methods that are outlined and that I like to talk about today are the DPRA, which is a peptide assay, the NRF2 pathway luciferase test, as well as the HCLAT test. And then finally, kind of a recommendation by the FDA within their guidance document on 10993 is that complaints associated with these three endpoints are analyzed and recorded at least annually. So again, the big three is kind of the cornerstone, the triangle of biocompatibility. So kind of extending on to the regulatory framework, we had a few questions about this. Is this aligned, is the in vitro testing that we're discussing aligned with the FDA efforts to reduce animal testing? So here's a little excerpt from the reference document, guidance document from the FDA, in saying that they agree with minimizing the number and exposure of test animals by giving preference to in vitro models where these models yield equally relevant information that are obtained from in vivo models. So what that's talking about is the industry kind of slogan, industry saying, an industry-wide effort to replace, reduce, and refine the use of animals within testing. So to kind of illustrate how these apply to them, I have a list here of the various endpoints, the in vitro models and the in vivo models that are closely related to them. So the first one being toxicity endpoint, as characterized by cytotoxicity. This is most closely related to systemic toxicity or implant effects, not directly related. Cytotoxicity is actually a really sensitive method And so it can be a leading indicator, of things to come when you scale up to the systemic toxicity or implant effects there. Irritation, the in vitro model is the RHE tissue model, reconstructed human epidermis tissue, and common in vivo model for that would be intracutaneous reactivity. And then finally, the sensitization endpoint. It's a whole battery of tests that's utilized to assess sensitization, which include, as I mentioned, the DPRA, NRF2 luciferase test, as well as HCLAT. And the in vivo models that those correspond to would be something like a guinea pig maximization test or local lymph node assay. So a similar question is, does in vitro testing serve for a 510K submission? And the one that does stand alone in a 510K submission would be cytotoxicity. With the other ones, all the other models, aiming to replace, reduce, or refine the animal testing that is required for an endpoint. So as far as direct guidance on the 510K submission, again, caveat is that that's up to your biological evaluation plan as defined by your toxicologist or biocompatibility specialist. But when it comes to the testing, these in vitro models can serve as a great screening method to test your product before scaling up into those larger, more expensive, time-consuming in vivo models that again have an impact on our animal population. So with that, I'd like to kind of get into a few of the actual in vitro assays and how they're conducted and give you a better understanding of how your materials may perform within them. So the first one, again, keeping with the same order, is in vitro cytotoxicity. So the way we start with cytotoxicity is that we have a cell culture of fibroblasts. So we use L929 fibroblasts in the study, and we culture them in tissue flasks. This is a little petri dish, which is usually used for bacteria. It's for information purposes only. We don't culture them, in petri dishes like this. So while we have those going and incubating and growing, we'll have our test articles. We'll prep them. And it could be a wristwatch, it could be headphones, it could be eyeglasses, and we'll perform an extraction in the same solvent that we actually grow the cells in. So the same culture media, the complete growth media that we grow the cells in. We'll then transfer those extracts to the cell cultures, and we'll run that cell culture alongside a negative control, so a non-cytotoxic extract, a positive control, which would be a cytotoxic extract, and a blank, so we call it the solvent control. We'll culture those, and then after 48 hours, we'll evaluate them under the microscope or by quantitative methods, and assign a score based on kind of the morphology and how healthy the cell culture appears. So when it comes to kind of defining, cytotoxicity study parameters, we can discuss time and temperature choices and how that differs with different device patient exposure. So the first parameter would be, your extract duration. First option for limited contact devices is generally a 24-hour extract period. The next one would be 72 hours, and that would be for products that have, prolonged contact or long-term contact. So anything 24 hours and above, so 24 to 30 days, 24 hours to 30 days, and then 30 days and beyond. And then in special conditions, maybe it's a material that's kind of in a transient contact, you could, rationalize a 4-hour extract period. Or if you want to look at it from a different angle, those are all things that we can evaluate. Finally, with temperature, the extracts are for in vitro, especially cytotoxicity, are always done at 37 degrees C. The reason for that is that higher temperatures can impact the chemistry in the extract solvent, which again is that growth media. So it's the same culture media that the cells are growing in, and if they're elevated at higher, or if they're extracted at higher temperatures, then you can get some interesting chemistry with that. Additionally, with the test article, so if you have a polymeric material and you extract above the glass transition temperature, you're going to have a lot more movement of the molecules, and you could over extract the compounds in a manner that's not mimicking physiological conditions or not mimicking clinically relevant or user relevant conditions. So for cytotoxicity and really most in vitro assays, we stick to that 37 degrees extraction time. So kind of to summarize that, we have our extraction conditions here, and then we will perform the extraction for 24 to 72 hours at 37 degrees C. We'll incubate them in the culture with the extract media, and then we will perform an evaluation on the back end, either by a qualitative evaluation by scoring them or a quantitative assay such as MTT. So now that we have discussed the cytotoxicity assay, I want to move on to in vitro irritation. And in vitro irritation, I kept this the same because the workflow of it is actually really similar. The main difference, other than some of the time changes are different, but for our purposes today, is that we're going to swap out the cell culture fibroblast for that reconstructed human epidermis tissue. And here's an image that's similar to what it's like, that kind of depicts the structure of that tissue culture. So this is a three-dimensional tissue culture that's grown. So this is looking a planar view. This is kind of the membrane that it's grown on, and then you see the different layers of the cells which mimic the epidermis tissue. We'll perform a similar extraction for 72 hours and then run it next to some controls. And then we'll perform an MTT assay. So this is a molecule that undergoes a redox reaction when the cells are metabolically active. So when the cells are healthy and living, they're going to go through this redox reaction, which will change the color of the dye to a purple. And then the percent absorbance is then correlated to the tissue viability of that tissue population within that group. And if the viability is less than 50% of the control, it would be considered an irritant. So I want to walk you through kind of what that would look like from an experimental sense. So this is kind of an overview of the internal validation that we ran on this assay. So we had some test articles that we actually made in-house. So one of them was a Y4 polymer, which is a PVC compound that is detailed in the ISO 10993 part 12, which is as a reference material. And then we had Y4 polymer, which is that same PVC base, along with Genopol X, which is an irritant chemical. So Y1 is expected to be a non-irritant, Y4 is expected to be an irritant. We ran that alongside some controls. So the negative control is saline, and then positive control is 1% SDS. And we ran an extraction on the test articles according to these conditions, so 72 hours and 37 degrees C, in both a polar extract, which was saline, and a non-polar extract, which was sesame oil. So these are the results that we received from that validation. And you can see here, this is the epidermis tissue overall viability plotted against the different groups that we had. The y-axis is percent viability normalized to the negative control, so it's at 100%. And you can see that our two vehicle controls, so the polar solvent and non-polar solvent, were both around that 100%, along with the negative control. We also see that our Y1 polymers were around that 100% in both the negative in the polar solvent as well as the non-polar solvent. And then we see our Y4 polymers, which we expected to be an irritant because of the chemical laced into them, were both irritants along with that positive control. And here is that 50% cutoff line to demonstrate that. So now that we've talked about cytotoxicity and irritation, I want to move on to in vitro sensitization. And this is an interesting one. We received some questions on the regulatory acceptance or what our experience is in using in vitro sensitization data to address the endpoint. And so to answer that, kind of want to look back at what is sensitization and what is the mechanism of it. So again, it's an immune response to a substance that reacts with skin proteins, rendering the substance immunogenic. So it's more of an immune response. And again, the ISO standard and the FDA recommend, using the in vitro methods for screening purposes prior to animal testing. So ultimately, the in vitro sensitization assay is used kind of for hazard identification before the in vivo studies were to be done. The reason for that is that it's a kind of a series of cascading events that the OECD has kind of categorized into 4 key events. So key event 1 is the protein or peptide binding. So that's the actual sensitizing chemical or sensitizing substance binding to the proteins within the cells to start that cascade. Key event 2, which follows up the protein binding, is keratinocyte activation, and that is the actual skin cells activating, beginning to produce detox enzymes to sort of help control that reaction. The third key event that's assessed in vitro is dendritic cell activation, and so this is getting more into the systemic immune response, leading up to key event 4, which is T cell proliferation, which is actually only assessed via in vivo testing. So that's kind of how it's broken down. This next slide, I want to kind of discuss a defined approach to the sensitization assay, because each key event on its own kind of stands, on its own. So a holistic approach is really necessary. So one of the common ones that's discussed in literature and in practice is the 2 out of 3 defined approach. And what that is doing, 1 assay for each of the first key events. So the DPRA, peptide binding assay, keratinosense, keratinocyte activation assay, key event 2, and then key event 3, the HCLAT dendritic activation assay, which is key event 3. So how it works is that you would conduct, any 2 of the assays, and if based on that, you look to see if the 2 results are concordant. If they are, then you have your conclusion that it's identified as a sensitizer or non-sensitizer hazard. If it is not, you would perform the 3rd assay and again see if they agree. If they do agree, you would have your categorized in if it's a sensitizer or non-sensitizer. And if not, then the assay would be inconclusive and further data would be needed. When we ask if these are concordant results, they can't be borderline. So if it's it's close to being a yes or a no, those cannot be necessarily considered. So it must be clear results. One thing that's really important to consider with this is what's the statistical validity of these studies. So I have this chart here that's summarizing the OECD data that's available in this reference here. And the criteria here are accuracy, sensitivity, specificity, and balanced accuracy. So accuracy is the correct classification rate. Sensitivity is the true positive rate. Specificity is the true negative rate. And then balanced accuracy is the average of sensitivity and specificity. So for the 2 of 3 approach against the local lymph node assay, which is a in vivo model, accuracy and sensitivity are at 83% and 82% respectively, and then specificity at 85% with balanced accuracy at 84%. For the 2 of 3 approach versus human, the numbers are a little bit higher at 89% for accuracy and sensitivity, and 88% for specificity and balanced accuracy. And then finally, for comparing the local lymph node assay versus human, which I think is important to put, kind of this first or I guess second column into context. The accuracy is 82%, sensitivity is really high at 94%, but specificity is at 22%, leading to the balanced accuracy being 58%. The reason the specificity is so low is that the true negative rate is low. So it misidentifies, it's common for it to misidentify, non-sensitizers as a sensitizer. So you get a high number of false positives, which is why, again, the sensitivity would be so high, is because it it doesn't miss as many. So that in a whole kind of shows the validity, the statistical significance of this approach compared to the human data and in vivo data. So now I'd like to take a little bit of time to kind of discuss, if the results are not as expected, where do we go from there? And I think that this is where EAG as a whole can really help. We can really leverage the resources within EAG as well as the Eurofins network. But the first thing that we would do is review the study design. Did we ask the right questions? Did we have the appropriate sample selected from the beginning? And did we prep the sample correctly? So we've worked with companies to isolate only specific regions of the test article. To maybe there's a sensor on one small little part of the product, and we only want to focus on that. Well, we can work with you to determine if there's a way to isolate that, or if there's a way that we can create a coupon that's going to present similar material properties when it comes to the biocompatibility of that product. The next step that we would look at is compare the results to a predicate product. So those of you who are in the medical device field are probably familiar with this, with the FDA 510K process, where if you have a failed cytotoxicity assay and you're running the product alongside of a predicate product that's known to be safe, there's clinical data to demonstrate its effectiveness, demonstrate its safety. You could run them alongside of each other to show that they have a similar response. The next thing that we can do is further analysis on the extract. So I bring up this chart here. So this is a smart chart that's actually available on our website, and it's interactive. So I encourage you all to visit our website and look at this. And it details not all of them, but the majority of the analytical techniques that we have in-house here at EAG. And it really emphasizes our ability to characterize bulk materials, but also products from 1 centimeter down to the nanometer scale. So these can be leveraged when doing a particle ID job. So maybe the extract process, we pulled out a bunch of particles and we need to actually understand what that is, because we suspect that led to the negative in vitro result. We can use the full gambit of testing that we have available. The next thing we could also look at is chemical characterization. So here in St. Louis, we have a full extractables and leachables team that specializes in ISO 10993 Part 18 testing, and that would do a full chemical identification of the product through LCMS or GCMS or other methods that can then be used to do a toxicological evaluation to determine the safety of the product. We could also look at surface chemistry. So you might notice a lot of the methods here, or the techniques in this smart chart, are surface chemistry. This would really be applicable in blood contacting devices. Maybe it is a true medical device that has a intracutaneous sensor. We could help by looking at the surface chemistry topography of the products. And then finally, as I mentioned earlier, we have a lot of experience in helping our clients source their materials. So there's numerous case studies where we have looked at, we've had a product fail, and it kind of kicks off a whole investigation to find that somewhere up the supply chain, the product is either produced on a similar manufacturing line, or combined with compounds that we don't want in the product. So those are all kind of approaches that we can take should something fail. So again, just to reiterate, kind of the industry that we work with would be medical device industry, consumer product industry, raw materials that are generally needing regulatory submission, design screening. We do a lot of as well as lot release. This is a chart that's actually available on our website that also kind of emphasizes the utilization by industry for in vitro testing. So, wearables and consumer products can utilize it for risk analysis, risk mitigation, marketing, not only to market that the product is safe, but also that no animals were tested when designing this product. They can be used for prototype evaluation and predictive modeling. For medical devices and biologics, we can do a lot of bioactivity assays for the products. So maybe you're working with osteoinductive hydrogel. We could design some assays to help with that, help characterize that. And then finally, also finished product in a bridge into in vivo testing or clinical trial testing. So in conclusion, we kind of walked through in vitro testing at EAG. We defined what a wearable is and what kind of differentiates a consumer product from a medical device when it comes to regulations. But ultimately, us as scientists, we're looking at the material hazards, and those exist regardless of some of the clinical data that is utilized or the clinical interpretation that's used. We discussed how testing suppliers and products can hedge against poor consumer experience. And then I outlined, the big three: biocompatibility, in vitro assays, cytotoxicity, sensitization, and irritation. And then finally, when it comes to failures, at EAG, we know how to test all materials. We want to help you gain a better understanding of your materials to deliver better products to patients and customers. So with that, I will hand it back to Melissa for Q&A.
Thank you, Blake, for the great presentation. As a reminder to our attendees, you can still submit questions through the control panel or your app. And now Blake and Philip will begin answering the questions submitted during today's presentation.
Perfect.
All right, thank you, Melissa. So looking over some of the questions that we've gotten so far, I'll just kind of start at the top and get your thoughts on these, Blake. So the first one's kind of talking about a more technical aspect of it. You mentioned using coupons for doing testing, and the question coming from the audience is, if a coupon approach is used in testing, how should that coupon be designed and prepared?
That's a good question. So when it comes to coupons, one, I think it's important to caveat that, a lot of these decisions should be made, within the context of your biological evaluation plan when it comes to a medical device. If you're not familiar with biological evaluation plans, it's basically, a gambit of testing that's defined ahead of time to, kind of indicate the pathway that you want to take in order to demonstrate that your product is going to be safe. So when it comes to coupons, I would heavily lean on your toxicologist team or your biocom specialist to help with that. But then when it comes to testing it, you want to make sure that the coupon is made in the same manner as your finished product, using the same materials and with the same processing. So if there's a sterilization method that's used, for, I'm thinking medical devices here, you want to make sure that that's retained there. And even down to some of the surface geometry, if you have a highly porous material that you run through EO sterilization and you don't let it off gas for more than 24 hours, and then you run cytotox on it, you might get some residual EO that's left in there. So you want to make sure that things are done in the same manner as your actual product.
Okay. Thanks. So
yeah.
Another question, something a little bit more on the project management planning side. This question asks that, including the extraction duration, how long would a cytotoxicity study take from sample submission to final report?
Yeah. So with everything, our standard kind of turnaround time is 2 to 3 weeks with that kind of testing. If we really need it done quickly, we can do it in the exact timeline that I kind of outlined. So, if it's 72-hour extraction and then 48 hours on the back end with the incubation, assuming that everything is in place and communication is done, properly ahead of time, we run all of our quality checks, all of our technical review in process, as well as technical review right at the very end. And so we kind of save a lot of time when it comes to that. So yeah, down to that 5 to 6 day turnaround time, actual days. When it comes to business days, do it over the weekend. We cut that down pretty quick.
All right, another technical question. To generate the error bars for in vitro testing, how many articles need to be extracted?
What was the first part of that? To generate the what?
The error bars. I think this is referencing back to the slide where you were discussing the validation. And you showed pretty good error bars where it's very clear that you see either it passes or fails.
Yeah. So with those, and I'd have to go back and look at the exact. I think that those were that was pulled from 6 replicates, but a lot of that was, again, it was really tight data, which kind of shows the robustness of that study. But across the board, most in vitro assays are going to be 3 replicates pulled from 1 sample. So for a cytotoxicity study, if we have something that is we're going to run it on a 3 centimeters squared per ml extract ratio. If we have 10, 15 centimeters squared of that product, and we select that extract ratio, then we'll use that 1 product, and then the extracts will be exposed to 3 cell populations, and so that's where you get the 3 replicates all from the one sample, and that's the industry standard for that.
Okay, so that probably helps with the number of devices needed for testing then.
Yeah, so we set it up. We set everything up in our lab for a lot of high throughput and to be the most cognizant of materials as we can, because I mean, a lot of these products are a lot more expensive than the test itself, and so the less that we can use, that helps out the clients, the better.
Okay. So some more questions kind of on the, maybe this is a broad question, just overall, maybe a little bit of a summary here. Why would one use in vitro testing instead of sensitizer screens or in vivo? So just kind of the big bullet points, I guess, is what they're looking for.
Yeah, and again, that goes back to the kind of predicting the in vivo response. If you've run these studies before, you know how expensive they are, and then also how time consuming they are. So you're looking on the order of up to 10 weeks for just a general sensitization study in vivo, and that's based on the different dosings that are done in that study, because you have an initial dose and a second dose and then a challenge dose, and those all have to be done with a certain amount of time elapsed in them. So there's just that limitation of biology and creating that response. So for sensitization, the biggest thing there is saving that time. If you can run the in vitro assay ahead of time to give yourself a better idea that it's going to pass that in vivo model, that can save a significant amount of time and resources on the back end, so that you don't have to backtrack and revise either the processing or the material, or maybe it was the wrong sample submitted or something. We wouldn't want to do that, but things like that do happen, and so testing early and often, especially for those high risk products, is going to save you time and money kind of on the back end.
All right. So following up on that statement about high versus low risk, we have another question that's asking about having multiple products, maybe in a similar family or maybe not.
Yeah.
But multiple products with different levels of risk, and then how do I go about setting up testing for these? Considering the time and cost and kind of balancing those.
Right. Yeah. So with that, I would probably look at for the higher risk products, and that could be risk with anything. Like it could be risk because the material is hard to source. It could be a risk in the sense that it is an actual hazardous risk that you're trying to process out. So with those, I would test early and often. If you're making a design change, maybe it's a somewhat unstable material, or it's a newer material, and you change up the formulation just a little bit. Just running a quick cytotox test, running a quick irritation or sensitization assay could, again, help you get that kind of leading indicator before you scale up that production. So yeah, running kind of those feasibility studies would really help. With the lower risk one, again, depends on how you define risk, but you can maybe test less often and later. So yeah, in summary, the high risk ones test early, often throughout the development process, and then maybe the lower risk ones that you're well aware of and can it's already well characterized. You can kind of put that testing off maybe towards the end.
Yeah, I guess that kind of dovetails too with FDA guidance on materials in contact with intact skin, right? And the need for in vitro testing, because I do know that there's the list, of course, of materials that are generally accepted to not be an issue, and so those clearly be counted as low risk, even though you still have that requirement to be reviewing yearly for around it.
Yeah, and again, going back to kind of the biological evaluation plan and your toxicology team or your biocomp team, those are kind of good questions to tackle there, and we're more than happy to help inform those from a testing standpoint too. So.
Okay, good. So another question, I think it follows well with the previous one, is how will this testing impact a product development timeline?
Yeah, I've kind of hit on it. I'm an engineer. I like testing. I like getting results. I like getting results early, and I like getting results fast. And so that's really kind of the big advantage there is having kind of that assurance moving forward before you scale up production, before you scale up into full in vivo studies or clinical trials with wearables. Testing that can save you a lot, and a lot of the tests will, again, it's a 1 week, 2 week, 3 week turnaround time, depending on what's going on in the lab and all of that, but it's something that we can get done fairly quickly to get you answers quicker to make better decisions.
All right, and then another one that's kind of further along in this process. Is this testing something that can help control supply chain?
Yeah, definitely. And again, it's from that raw material selection side. So are the polymers that you're using, are the adhesives that you're using, are the different components that you're using? Is the stainless steel really stainless steel? Or is there a lot of nickel? I mean, it'll have nickel in there, but what are the tolerances there? And those are things that a cytotox screen can get you that answer, in just a week or two.
All right. And then let's see, looking over, let's do one more. So this last question is just kind of asking what the typical turnaround time for the big three testing in vitro is compared to in vivo. So kind of giving an idea of like what's the magnitude of that that difference in time. Yeah.
So like I mentioned, cytotoxicity is 2-3 weeks is standard for and with the right communication and we can rush it faster than that. With the irritation assay, it's a little bit different, where that tissue is actually grown to order, and so that's looking at closer to that 3 or 4 week turnaround time. But also, if it's something that we know the samples will arrive and we can kind of work ahead and we have assurance that the tissues won't go to waste, we can kind of maybe get those ordered ahead of time to help coordinate there. So it really, my big emphasis is just on communication ahead of time with project managers, and that kind of goes with anything. Setting the right expectations, and if we have the right communication and are all on the same page, then we can do a lot for you. And then sensitization, those assays are still kind of in development here at EAG, but in terms of like the biological mechanism of them and the logistics of running the assay, it'll be similar to that cytotoxicity turnaround time for 2 to 3 weeks.
All right, good, thank you.
Yeah.
So we'll wrap it up here, Melissa. So I'll hand it back to you.
Perfect. Thanks, Philip.
All right. Thank you, Blake and Philip, and thank you all for attending today's webinar. If we didn't get to your question today, we will follow up with you soon, and we will also send you a recording of this webinar. And if you have any other questions, please send them to customerservice@eurofins.eag.com. It's on your screen. And once you leave today's webinar, you'll receive a survey on the presentation, and we would really appreciate your feedback. And on behalf of Eurofins EAG Laboratories and our experts, thank you for joining us and have a wonderful afternoon.
Thanks, everyone.